Good evening, everybody. I'm Dr. Bobby. Welcome back, all of you, on behalf of New Age Cosmetic White Edtech Health Consultancy. I'm on to Unit 4 of correction. We're on to Unit 13 of the CSIR National Eligibility Test Life Sciences syllabus, which covers the subject of recombinant DNA technology, which comes under the title of Methods in Biology. On to the subject today. I welcome all of you to please take notes and necessary screenshots of figures that would be shown during the course of the lecture. Right, and now starting on with the subject matter proper, recombinant DNA technology. The general info would include A, landmarks in modern genetics. One would be rediscovery of Mendel's works. 2 would be chromosomal theory of inheritance. 3, DNA as the genetic material. 4 would be recombinant DNA technology, development and applications. That will be followed by B, recombinant DNA refers to the creation of new combinations of DNA segments that are not found together in nature. The isolation and manipulation of genes allows for more precise genetic analysis as well as practical applications in medicine, agriculture, and industry. Fundamental changes in our society are occurring as a result of genetic engineering. There's a figure down there. Please take a screenshot as you may for the benefit of your studies. Second will be making recombinant DNA. Overview. Isolate DNA leads to cut with restriction enzymes, leads to ligate into cloning vector, then to transform recombinant DNA molecule into host cell, and then onto each transformed cell will divide many times to form a colony of millions of cells, each of which carries the recombinant DNA molecule DNA clone. Now this figure is self-explanatory. I'm just magnifying it for your understanding of it. There's the biological process A under study, which identifies a gene by classical genetics. So we have extract DNA from organism or extract vector from bacteria, then cut DNA into fragments, you open the vector, make a recombinant DNA, introduction into bacteria to clone and select clone with gene A. So take this diagram down, self-explanatory for you to explain your answers during your exams. And from this is courtesy from an introduction to genetic analysis by 6E by Griffiths, Miller, Suzuki, Leonton, Gibbard, copyrighted in 1996 by W.H. Freeman and Company, used with permission. Right? So all credits to them for this beautiful illustration. Now, A, isolating DNA. Crude isolation of donor foreign DNA is accomplished by isolating cells, disrupting lipid membranes with detergents, destroying proteins with phenol or proteases, degrading RNAs with RNAs, and leaving DNA at the end. The next will be crude isolation of plasmid vector DNA, which is accomplished by an alkaline lysis procedure of a boiling cells, which removes bacterial chromosomal DNA from plasmid DNA. 3 would be to get pure DNA from either 1 or 2 crude DNA is A, fractionated on a CaCl2 gradient, and precipitated with ethanol being the second part, and third would be poured over a resin column that specifically binds DNA. Cutting DNA. 1 would be DNA can be cut into large fragments by mechanical shearing. 2 is restriction enzymes are the scissors of molecular genetics. Restriction enzymes, RE, are endonucleases that will recognize specific nucleotide sequences in the DNA and break the DNA chain at those points. A variety of RE, that's restriction enzymes, have been isolated and are commercially available. Most cut at specific palindromic sites in the DNA sequence that is the same or both antiparallel DNA strands. These cuts can be staggered with generate sticky or overhanging edge, or blunt, which generate flush ends. So these are the genetic coding. You could just about make a note of that. C would be joining DNA. Once you have isolated and cut the donor and vector DNAs, they must be joined together. Let me just de-magnify it, reduce the magnification so I get the sentences right. Right, and the excuse me, the DNAs are mixed together in a tube. If both have been cut with the same RE, that is restriction enzyme, the ends will match up because they're sticky. DNA ligase is the glue of molecular genetics that holds the ends of the DNAs together. DNA ligase creates a phosphodiester bond between two DNA ends. This figure shows that we have a plasmid vector with a cleavage site, and cleavage is by E. KORAH endonucleates, and foreigner DNA cleavage sites, and the cleavage by E. KORAH endonucleases, and that goes to annealing with DNA ligase given a plasmid chimera. This is from an introduction to genetic analysis by 6E by Griffiths, Miller, Suzuki, Leonson, Gerbot, copyrighted 1996 by W.H. Freeman and Company, used with permission. Credits to all of them, and thank you so much. D. Amplifying the recombinant DNA. To recover large amounts of the DNA molecule, recombinant DNA molecule, it must be amplified. This is accomplished by transforming the recombinant DNA into a bacterial host strain. The cells are treated with CaCl2 to give DNA and is added. Cells are heat shocked at 42 degrees centigrade, and DNA goes into cell by a somewhat unknown mechanism. Once in a cell, the recombinant DNA will be replicated. When the cell divides, the replicated recombinant molecules go to both daughter cells, which stem cells will divide later. Thus, the DNA is amplified. You have the target sites, foreign DNA, after cleavage, DNA entering the vector. They transform bacteria, which is replication, host chromosomes, cell division, which is amplified to give the newly formed clone. Well, this again is an introduction by the from the text Introduction to Genetic Analysis by Griffiths, Miller, Suzuki, Leonson, and Gerbot by 1996, copyrighted W.H. Freeman and Company, used with permission. Thank you. Credits to them. About a DNA clone, a section of DNA that has been inserted into a vector molecule and then replicated in a host cell to form many copies. Vectors. Requirements for a cloning vector. Should be capable of replicating a host cell, should have convenient RE sites for inserting DNA of interest, should have a selectable marker to indicate which host cell receives a recombinant DNA molecule, should be small and easy to isolate. Going on to the second part. Bacterial plasmids are small, circular DNA molecules that are separate from the rest of the chromosome. They replicate independently of the bacterial chromosome, useful for decloning DNA. Inserts less than 20 kb kilobase pairs. Inserts larger than 20 kilobase are lost easily in the bacterial cell. So please note this figure here. And this, take screenshots for your references and make notes. Kindly, please do not commercialize any aspects of these lectures. It's only for study purpose. From Introduction to Genetic Analysis by 6E by Griffiths, Miller, Suzuki, Leonson, and Gerbot at 1996 by W.H. Freeman and Company, used with permission. Credits to them. Thank you. 3. Bacteriophage lambda 45 kb contains a central region of 15 kb that is not required for replication or formation of progeny phage in E. coli. Thus, lambda can be used as a cloning vector by replacing the central 15 kilobase pairs with 10 to 15 kb of foreign DNA. This is done as follows. Mix RE, restriction enzyme, cut donor DNA and lambda DNA in a test tube. Ligate, use in vitro packaging mix that would assemble progeny phage carrying the foreign DNA. Infect E. coli with pair phage to amplify. This is the figure of the amplification and the cloning. And the genomic DNA, which goes on to SAU3A sites, so partial digest with SAU3A. A cleave is a subset, And there you have 45 kilobytes of BAMHI, and that goes to bacteriophage lambda vector. And finally, after cleavage, in vitro cleavage of concatemma, packaged into phages, and we have plaques that screen library used nucleic acid probe. And this is an explanation of the procedure, an introduction to genetic analysis. Credits to all of the authors. Thank you. And 4. Cosmids are hybrids of phages and plasmids that carry DNA fragments up to 45 kb. They can replicate like plasmids but can be packaged like phage lambda. All right. This is another figure. Please take note. To keep a screenshot. Credits to the introduction of genetic analysis by the authors of and copyrighted at W.H. Freeman and Company, who used permission. Thank you. Expression vectors are vectors that carry host signals that facilitate the transcription and translation of an inserted gene. They are very useful for expressing eukaryotic genes in bacteria. 6. Yeast artificial chromosomes, YACS, are yeast vectors that have been engineered to contain a centromere, telomere, a region of replication, and a selectable marker. They can carry up to 1,000 kb of DNA. Since they are maintained in yeast, a eukaryote, they are useful for cloning eukaryote genes that contain introns. Also, eukaryotic genes are more easily expressed in a eukaryotic host such as yeast. 7. Bacterial artificial chromosomes, BACs, are bacterial plasmids derived from the F plasmid. They are capable of carrying up to 300 kb of DNA. And now making a library. A library is a collection of DNA clones in a certain vector. The goal is to have each gene represented in the library at least once. B. Types are categorized by 1. Source of vector DNA, 2. Source of donor DNA, A. 2A, that's a genomic made from the restriction enzyme DNA fragments, a total genomic DNA. B. Chromosome made from RE DNA fragments, or one chromosome isolated via flow cytometry or pulsed field gel electrophoresis. C. cDNA, that's complementary DNA made from DNA synthesized from mRNA. No need to worry about introns. Note these diagrams and the credits to W.H. Freeman and Company for the authors of An Introduction to Genetic Analysis. Now, a genetic identification of the specific gene of interest in the library. A. Probing for the gene. D. 1. DNA probe. A. DNA probes are based on the fact that a DNA should heated or chemically heated, treated to become single-stranded DNA molecule will hybridize, bind to sequences that match or are similar to it. B. Where does the probe DNA come from? 1. The cDNA from highly expressed mRNA from a tissue, homologous gene from a related organism. 3. DNA obtained from reverse genetics, protein DNA. If you have the protein product of the gene in which you are interested, sequence part of the protein, synthesize a short greater than 20 nucleotides, DNA probe based the protein sequence using the genetic code, use your probe. And now let's take this diagram for your reference. Right, we see cellular genome, cleavage with restriction enzyme. We have bacteriophage, extract DNA, cleave. A nitrocellulose filter, incubate filter with a radioactive probe. And that leads to hybrid phages, along with, and there's a ligation in the presence of recombinant DNA, which it comes from after cleavage of extract DNA from a bacteriophage. Then that moves on to infection of the bacterium, lysis and plaque, and the lawn of bacterium, which finally leads to a phage clone plaque, a library of phage clones. Courtesy to the authors who've been copyrighted and produced by W.H. Freeman and Company, use with permission, credits to them. 2 is protein probe. If you have the protein product, the gene of interest, make an antibody against it. Use the antibody to protein of interest is used to screen the library for the clone that is expressing the gene that codes for the protein. Please note the diagram. It's self-explanatory. You have the overlay of nitrocellulose filter into the fusion protein in vitro packaging plate on bacterial lawn. And where the protein binds to a nitrocellulose. Finally, you have the autoradiography onto the X-ray film, antibodies identifying specific blocks, primary antibody, and the fusion protein bound to nitrocellulose, courtesy Introduction to Genetic Analysis by Freeman and Company. Thank you. Next is the complementation B.